Hi, my name is Barney Whitman, and I'll be providing an overview of the International Code of Nomenclature of Prokaryotes described from sequence data. And here's my dog Shep, who will be joining us. This talk is an overview of the online document entitled Introduction and Draft of the SeqCode, which provides a detailed discussion as well as the actual code. This code is informally called the SeqCode to distinguish it from the ICNP, or just the code. This name emphasizes that it uses DNA sequences as a nomenclatural type, while the ICNP requires deposition of type strains in 2 culture collections. It's also been informally called the uncode to emphasize that a major motivation, i.e. to provide permanent names for uncultivated prokaryotes. However, the SeqCode can also be used for naming deciduous organisms and symbionts that cannot be deposited in culture collections, as well as organisms that cannot be removed from their country of isolation due to legalities such as the Nagoya Protocol. So why do we need names? Single precise names of prokaryotes are essential for efficient communication in biological as well as other sciences. Stable names are also necessary for creation of large databases that enable complex computational analysis. So without stable names, names would have to be frequently curated, which would become increasingly difficult as the databases get larger and larger. How do codes create stable names? Codes of nomenclature establish identifiers, called names, that have a one-to-one correspondence with natural entities by associating names with types. The type, which is the evidence for the biological entity, which would be a DNA sequence in the SeqCode, or it could be a subordinate taxon, like a species is a type for a genus. Stability is achieved by the principle of priority, i.e. the earliest name that encompasses the type must be used. Why use sequence data as type? A major purpose of the nomenclatural type is to provide a standard for comparison of new specimens to determine whether or not they belong to the same or different taxa. A sequence can be easily compared. For instance, the average nucleotide identity, or ANI, between 2 genome sequences can be easily calculated to determine whether or not they should be classified in the same species. A sequence can be easily shared and downloaded from databases. Lastly, a sequence provides a permanent record. For instance, the oldest books are probably about 4,000 years old. In contrast, there are very few bacterial cultures which have been maintained in culture collections for more than 100 years. And so whether or not a strain can be kept for millennia is still unproven. So how does the SeqCode work? Basically the same as the ICNP. The rules for forming names are the same. Names have priority and cannot be changed unless they are found to violate the code. And the hierarchical taxonomic ranks are very similar. In the SeqCode, a registration system will be established so that names can be validly published, just like in the ICNP. We also anticipate the establishment of 2 committees to administer the registration, updates of the code, and deciding ambiguous applications of the code. And these would be similar to the International Committee on the Systematics of Prokaryotes and its Judicial Commission. Priority for names created under the SeqCode will begin on January 1st, 2022. Names formed under the ICNP before that date will have priority in the SeqCode as well. After that date, names formed under the SeqCode and the ICNP will compete for priority. Because the SeqCode incorporates names formed under the ICNP, it allows creation of a uniform nomenclature for both cultivated and uncultivated prokaryotes. And it breaks down barriers between disciplines and enables numerous synergies within all fields of microbiology. This advantage of the SeqCode is difficult to overstate, and we think this will be a tremendous advantage to all areas of microbiology going forward. Like the ICNP, the SeqCode strives to be philosophically neutral on the great issues of systematics. For instance, it doesn't say what a species is or the meaning of any of the higher taxa. While it allows creation of hierarchical taxonomy, it does not specify the circumscription of ranks or any particular taxonomy. And this is a great advantage because as taxonomic philosophy has changed over time, it allows the names to remain constant. So what about Candidatus names? The ICNP currently allows Candidatus names for organisms described on the basis of their DNA sequences. Because these names do not have priority, they are not stable. Thus, the Candidatus names do not meet one of the major requirements of the SeqCode, i.e. to create stable names for uncultivated organisms. If you're interested in discussing this topic further, please join Breakout Group 4 entitled, Should the SeqCode Include a Proposal to Create an Approved List for Candidatus Names? There are some differences between the SeqCode and the ICNP. In both codes, the names of the higher taxa, this would be family and above, are formed from the names of a subordinate genus. However, the ICNP does not provide clear rules for priority of higher taxa. The SeqCode recommends that the priority of the higher taxa depends on the priority of the type genus. The advantages of this system become obvious when we discuss the following example. So consider a family that contains two genera, genus 1 and genus 2, where genus 1 has priority over genus 2. This means that the name of the family will be based upon genus 1, as will the name of the order and the class. There's also a second taxon, genus 3, that's a member of a family named after genus 3, family 3, and then order 3 and class 3. If we move genus 2 from family 1 to family 3, the codes require different things. Under the new rules, the family now becomes named after the genus with priority, so it's now family 2, order 2, and class 2 contain genus 2 and genus 3. Under the ICPN rules, this family retains its name of family 3, even though it includes genus 2 with higher priority. Now, if we take this new taxa, so family 2 contains genus 2 and genus 3, and now we unite genus 2 and 3. Because genus 2 has priority, the new taxon is genus 2, which is a member of family 2, order 2, and class 2. However, under the ICNP, if we unite genus 2 and genus 3, the name of the new genus now is genus 2, but now it's in family 3, order 3, and class 3, even though genus 3 no longer exists. If you're interested in this topic, please join Breakout Group 3 entitled Should the SeqCode Specify that the Priority of Higher Taxa Depend upon the Priority of the Genus and Species? Another difference is that the taxonomic ranks with standing and nomenclature in the SeqCode are reduced, and the ranks of subgenus, subtribe, tribe, subfamily, suborder, and subclass are not included in the SeqCode. These ranks are seldom used in the modern literature, and their experimental basis is ambiguous. The SeqCode also reduces the number of rules from 65 to 51 by combining similar rules and eliminating redundancies and rules no longer needed after creation of the approved list. I'd also like to acknowledge the co-editors for the SeqCode, and in particular, Brian Hedlund and Anna Louise Reisenbach, who obtained funding from the National Science Foundation for this workshop, as well as 2 previous workshops held in 2019 on the same topic. Also, we should recognize Allison Murray, who was the corresponding author on the roadmap for the naming of uncultivated archaea and bacteria that was published last year. Lastly, I'd also like to thank Kostas Konstantinidis, Ramon Rosella Mora, and Ian Sutcliffe, who worked on this project with me in the ICSP for many years. So thanks for your attention, and here's some further reading on this topic if you're interested.